the messy chemistry and the origin of life. And we're starting off now with Albert, and he's going to be talking to us about a continuous reaction network that produces RNA precursors. So whenever you're ready, Albert, take it away.
All right, thank you, Luke. Can you guys all hear me and see me, I guess? Just making
Yep,
sure.
you're coming through perfectly.
Great. Hello, everybody. My name is Dr. Albert Fahrenbach. Just call me Albert. I'm from UNSW School of Chemistry, and I'm a chemist. I do a lot of prebiotic chemistry. And today I'm going to talk to you about a reaction network that can produce precursors that are known to be useful for making RNA abiotically. And so let's get going. Luke says if I go over time, he's going to sound the air horn on me. So I don't want to be late. So here's something that we do in my group, some stuff. So like I said just before, this is going to be a talk about reaction networks involving small molecules. And the idea is we want to mimic chemistry that might give us an idea of what a proto-metabolism might have looked like on the early Earth. And some other stuff that we do I won't talk about today is RNA synthesis. And our main goal there is try to understand fundamental interactions between RNA and peptides, which is of course important for origin of life chemistry. And in collaboration with Martin's group and Luke, the chair, we're looking at some of these non-enzymatic RNA polymerization reactions, so-called ground truthing experiments, to kind of put them in a more realistic geological context. So anyway, so in a nutshell, you know, I'm sure people have seen this slide before. This is explains what I'm interested in, and at the same time, the entire history of the universe as well. So you have the Big Bang in this left-hand corner, and we have all sorts of interesting phenomena happening: galaxy evolution, solar system formation. Somewhere around here is the formation of the Earth. Very different place, as you can see. But then at some point, there is some transition that brought about this origin of life, and after that thing was about Darwinian evolution. So of course, we're interested in what exactly happened at this point. And we have reason to believe that it was probably pretty messy, right? Pretty complex types of reactions going on. And just to give you a metaphor for that sort of chemistry, I'm going to tell you a little anecdote about the so-called Library of Babel. So the Library of Babel was a short story written by Jorge Luis Borges. And the Library of Babel is a library that contains all possible books that could ever be written in a given alphabet. So it's the whole idea of like monkeys on a typewriter, right? You just let them go and they just mostly write nonsense. And that's exactly what the Library of Babel has, a lot of nonsense. But every once in a while, you might come across a book, like one of the works of Shakespeare, for example. Every science paper ever written has to be in this library. And it even contains the story of how life started on Earth. But in between all those valuable pieces of information, it's mostly just complete garbage. You know, and somebody actually went online and made a digital version of this Library of Babel. This volume, opened up a book, and what do you know, I found the my exact abstract for this talk written down in that library already. Of course, I never actually found that. I just, I searched it. They had a searching algorithm. You would never be able to find this in reality because the library is too vast. And in between, anything valuable is just garbage. Like here's the previous page. Here's the following page where my abstract was hiding. So this is kind of how chemists in general tend to view complexity. So is prebiotic chemistry like searching through a vast chemical library of Babel, where every atom is a possible letter, every molecule a possible word, every reaction a possible sentence, every network of reactions a possible paragraph, every geochemistry a possible page, and every exoplanet a possible book, with the vast majority producing only Babel? So chemists, because of this, they're really afraid to get their hands dirty, so to speak, with all this complex chemistry, because they're, you know, worried they're never going to figure it out. And for this reason, and historically speaking, chemistry community as a whole has tried to avoid complexity. That's just how the historically chemistry has been brought up. We've been taught to minimize complex reaction mixtures as much as possible, focusing on so-called target-directed synthesis. Prebiotic chemists have to begin embracing complexity, however, if we're ever going to begin sifting through this vast chemical library of Babel. So the story I'm going to talk about for the rest of my time is a story about complex chemistry that starts from relatively simple beginnings. And so where are we going to start? Well, how about a warm little pond, you know? And one thing that we're sure of, pretty sure, I mean, there's always doubt, but if you had dry land, then you would have had water that formed small reservoirs of, aqueous conditions and these small ponds or whatever would have evaporated periodically and maybe re-wetted from time to time, right? So with that said, this chemistry I'm going to talk to you about is relevant to this sort of scenario as opposed to somewhere deep in the ocean. Not that I think somewhere deep in the ocean isn't relevant, but you got to focus on something. And what are we going to use for our materials? Well, we're going to start with HCN, hydrogen cyanide. And so hydrogen cyanide is easy to make, so to speak. You can get it from spark discharges, like lightning. You can get it from space and then have it delivered to Earth, for example. Excuse me. You can form it through impacts when you have asteroids impact the Earth, and you can actually make it during that process. And so it may have been a common feedstock on the early Earth, and I'm certainly not the only person who has used cyanide in their prebiotic experiments. So we're also just going to add a little something else to the mixture. We're going to add some ammonia. We're going to add salt, actually a lot of salt. We're going to add a little bit of phosphate because we'll eventually need that to make RNA or something like that eventually. And of course, we're going to have water as well. What about energy? So life is non-equilibrium system, right? We got to have some sort of energy input to drive the system to do interesting stuff. So what we're going to use is mostly in the form of radical chemistry. That's where we're going to get our energy. We're going to make these radicals by ionizing either water or chloride. And in the process, we're going to make solvated electrons. We're going to kick an electron out of water, out of chloride. And in that, the byproduct is going to be OH radical, for example, in the case of water, and Cl radical, chlorine radical, in the case of chloride. And these two things on the left and right are powerful oxidants, whereas the one in the middle, that solvated electron, is a powerful reductant. And there's a variety of different ways you can actually get these things just by different forms of radiation, basically. And I'm going to talk to you today about gamma rays. So we've been using gamma rays to do this type of chemistry. So the basic idea is, could we start from these simple molecules, some organic and inorganic species? We zap it with some energy. We get these mixtures to complexify, and then maybe we can carry out some dry wet cycles or wet dry cycles, and at the end of the day, make something like RNA. And of course, this is following on the heels of the RNA world hypothesis, which I think this audience already knows about. So here's some of the team members that have been involved. I got to thank Reed Chan most of all because he's done most of the work that you're about to see. He's a member of ELSI at the moment in the US. So gamma rays, this is the gamma ray facility, and these are some of the vials that we would irradiate. So it's actually convenient to work with gamma rays. You just put these vials around the cobalt-60 source, and the gamma rays can pass through anything, and they can easily pass through the glass of the vials, irradiate your stuff inside. And recently, we've been doing stuff in at the radiolysis facility in Australia, INTO. So here's the basic idea. We're going to use water, some salt. That's going to make these oxidizing and reducing mixtures at the same time. That's going to be subjected to HCN, and maybe we can get all the way to the ribonucleotides. And basically, we're following the Pounder-Sutherland pathway that was set out or described by the researchers Pounder and Sutherland. And so the basic idea is that we were thinking when we first started this project is, can we get to this intermediate here, this compound in red? Because it's kind of like halfway en route to this molecule for the pyrimidine ribonucleotides. So here's the experiment that we did. So we took some cyanide, some salt, ammonium phosphate, just blasted it with some gamma rays. We end up making both these oxidized and reduced compounds. We do a wet dry cycle that frees up some of this compound on the left, known as glyceronitrile. It frees it up, gets rid of this nitrile to make glycoaldehyde, and then these now can react, and we can get our next intermediate in that pathway. And here's a little bit how that works. You know, when we dry it down, we just get rid of this HCN, and that's how we can generate this free glycoaldehyde needed for the reaction. Here's what the actual data looks like if you're interested. I won't go belabor the point too much, but you can see, you know, this is an NMR spectrum right after we do the radiolysis, and then we dry it down and heat it up for a little bit. We can see these peaks start to grow in. So if we zoom out of that NMR spectrum, you can also see this is in the presence of a pretty complex mixture, right? We're also making a lot of stuff at the same time. Some of this stuff was also useful for making amino acids, like aminoacetylnitrile, for example. And you have things like formamide, which is well known for different prebiotic transformations. So the point here is, you know, we really didn't have to do much, and we can get about 50% of the way to RNA after a day, you know? Very minimal human intervention. We just let the reaction do what it does, and we don't try to minimize any sort of complexity. So here is what the overall reaction network looks like. This is what we think is going on. Starting from HCN, we can get this reductive side of the reaction network going. That makes things like these aldehydes, which resemble simple sugars. On the right-hand side, we have this oxidation chemistry going on, and that allows us to make this compound in red in particular. The next steps, if we wanted to keep taking this reaction further, would be to make the next sugar in the synthesis that we need, which is glyceraldehyde. If we can get that to react with the amino oxazole, we get the next intermediate, and with a little luck, we can get all the way to the ribonucleotides. So this is a work in progress. There's no reason that we found so far that we couldn't actually do this. So some more work in progress. I just want to give some plugs to my student Jeremy Tran and Renee Lim, who they spoke yesterday. So Jeremy is interested in moving away from hydrogen cyanide as well as gamma radiation, and he wants to look into different feedstocks like formaldehyde and glycine that can take the place of HCN. Because if you could do that, you could make your reaction network more robust. And he's also looking into using UV light as a way to make these radicals. And Renee is also studying radicals computationally now, looking at how this different mechanisms for cyanamide can form. And with my colleague, Zach Adam at the University of Arizona, we've started looking at reaction network analysis of these complicated radiolysis mixtures. So we've combed the database of possible radiolysis reactions and started putting them together in a network in order to see if they have sort of lifelike properties with respect to network. So take-home messages, we should try to embrace. Hello, can you hear me? My internet went out. Can you still hear me? Is it all good?
Oop, we can hear you now, Albert. Yep.
Okay.
Good.
I'm not sure how long that went out, but so take-home messages. We should try to embrace complex chemistry, right? That's the only thing you get from my talk. That'd be a good thing. We have to move away from these kind of simple reactions and just go for it. And this may be the only way to eventually make sense of this chemical library of Babel that we're faced with. And please reach out if you'd like to collaborate. So with that said, again, I'd just like to thank Reach In mostly for doing most of the work that you saw, and Jeremy as well, and Renee, and all of you for your attention.
Amazing. Thank you so much, Albert. That was a great talk. It looks like we've got like a minute to spare quickly, but there aren't any questions specific for your talk. I think Harold's got some questions about his previous work. So maybe we'll just sort of keep pushing on and go to our next talk, if that's okay with everybody. So our next talk now is from Rowena Ball. and they're presenting on an anomalous thermal fluctuation distribution favors primordial chemical evolution. So, Rowena, are you you're there? Could you turn your microphone on and have a chat to see if you're present?
Hi. So
you're coming through a bit faint. Is there any way you could either get closer to the computer or turn your microphone up, potentially?
is that better?
Yeah, I think that's fine. Yeah, I think everyone should be able to just turn up their headphones if they need to. So yeah, if you want to just take your slides full screen so we can all just see them in presentation mode, then we'll be good to go.
So right.
That's fantastic. Thank you so much.
Great. So it's really great to be contributing here. My partner in crime for this work is John Brindley at Leeds University. And I'm really only going to say two things in this talk. Firstly, describe how our investigations into fluctuations, specifically a fluctuating periodic drive, can structure a complex chemical system, and how the results of those investigations led us to revise a necessary condition for the origin and persistence of life. Now, we've heard a lot of really cool stuff about the chemical requirements for the origin of life, and planetary requirements and geochemical requirements. We've been looking at the dynamical requirements. So it's usually, in fact always, posited as a necessary but not sufficient condition that the environment must be far from equilibrium. So we can't talk about near equilibrium or even near equilibrium environments. Thermal periodicity. So it's been very interesting hearing about the wet-dry cycles. We use thermal cycles to drive our chemistry. So heating and cooling cycles for non-enzymic RNA replication and biopolymerization and proto-metabolism. So what about, we've just heard about from Albert about the complexity, how we should embrace complexity and the messiness of the primordial origin of life environment, stochasticity. So environmental fluctuations are inevitable. And that led us to ask whether fluctuations are actually necessary for complex chemical evolution to occur. And looking at the literature, there seems to be some recent consensus that that is in fact the case. So here's one quote: a pre-biological system able to adapt to variations, or in fact make use of them, would be more resilient. So from 2019, and another one from 2017. Fluctuations are everything. Okay. And I'll just say a little bit about the open flow cell model that we use to simulate our fluctuating environment driven by periodic thermal cycles. So here we have an open cell, and it has an input, a mass input, and mass output. So it's an open flow through cell. And because it's an open thermodynamic system, it's sitting in a thermal bath at an ambient temperature that is randomly fluctuating. And to the input of our open flow cell, we add hydrogen peroxide and a bit of acid and thiosulfate. And this is a well-known thermochemical oscillator. So inside the cell, we get temperature oscillation. So in the input, we now add reactants that are thought to be important in prebiotic chemistry, such as pyruvate and glyoxylate for proto-metabolic cycles, some sort of surfactant or amphiphilic species that can potentially form vesicles, micelles, short peptides, and polynucleotides, not necessarily all at the same time. And under the influence of the thermally periodic drive, inside the cell, we get metabolic cycles and polymerization and vesicle formation. Now, as I mentioned before, the cell's sitting in a thermal bath at a randomly fluctuating ambient temperature. So this provides the random fluctuations of the input. And because it's a random fluctuation, it's a normally distributed or Gaussian fluctuation. What we asked is, that's the input fluctuation, what do the output fluctuations look like? Now this really floored us. This is the output fluctuation distribution, thermal fluctuation distribution. And the first thing we notice is that Gaussian perturbations of the inputs do not result in Gaussian perturbations of the outputs. And that's not obvious. And what we have, the second thing we notice is that what we have is this bimodal distribution. On the left, we have the steady state contribution to the fluctuation distribution. And on the right, this smaller hump, the smaller hump on the right, we have the dynamical contribution. So the fluctuations in the amplitude of the thermal oscillations. So we disentangled those two contributions to look at them. So here we have the steady state contribution on its own. And we see again that the Gaussian perturbation, so the normal distribution of the perturbations to the input, haven't resulted in a normally distributed output. And in fact, the distribution is weighted towards lower temperatures so that large temperature fluctuations occur with relatively low frequency. And what this means is that we're not getting enough high temperature fluctuations to drive the high activation energy reactions you need to sustain metabolic cycles and biosynthesis and chemical evolution. So we more frequently get low activation energy reactions, hydrolysis and degradation. So what does the dynamical contribution look like? We found that with the dynamical contribution, so the fluctuations in amplitude of the thermal oscillations, the distribution is weighted towards higher temperatures, and large temperature fluctuations occur with high frequency. So that high activation energy synthetic reactions are favoured over the low activation energy, the reverse hydrolysis and degradations. And so what we have is that complex processes leading to life are enabled and sustained. And I'll just go to a single example of peptide stabilized vesicles. So here we have our non-equilibrium open flow through cell in which we've set up the thermal oscillation using hydrogen peroxide and thiosulfate. And we've just added a single reactant. We've just added this surfactant, this amphiphilic lipid type species that can potentially form membranes. And that's what it does. It forms vesicles. So here on the top row, we have this simulated experiment with the fluctuations, the input fluctuations turned off. And we look at the concentration of vesicles, which is normalized to 1. Then we turn the fluctuations on this second row. And we find that the fluctuations make little difference. So with a single reaction, the fluctuations make little difference on average because the vesicles can form and then under thermal stress, they can degrade again, but all they can do is form again. The picture changes quite significantly when more reactants are added. So here we've added short peptides, which inside the cell form activated peptides, which then are incorporated or condense with the surfactant to form peptide stabilized vesicles. So here we have a table in which in the top line, the input thermal fluctuation is turned off and all the concentrations are stabilized to and normalized to 1. And on the second line, we look at the effect of input fluctuations with the several reactants. So what we find is that the rare high temperature fluctuations allow the high activation energy reaction short peptide to activated peptide to occur. The concentration of active peptide is lower because it more rapidly partitions into the peptide vesicles. The concentration of simple vesicles, V, declines because the monomers more rapidly form the stabilized peptide vesicles. And the peptide vesicle concentration increases quite dramatically. So what does all this mean? Okay. The benefits of thermal fluctuations to incipient life in these simulated experiments is that a fluctuating system favours complex over simple chemical systems. And secondly, a fluctuating system allows complex stable structures to prevail over unstable structures such as pure lipid vesicles. Secondly, net biosynthesis may occur under a fluctuating dynamical drive. So on the right, we have the fluctuation distribution from the fluctuating dynamical drive, but eventually must fail in fluctuating steady state. So on the left, we have the life-denying scenario of a steady state fluctuating situation. Although both situations are far from equilibrium. And that leads us to revise the necessary condition for life to originate and persist, and recast it as dynamically far from equilibrium. The guts of this research was published earlier this year in PCCP. I can provide that reference, a link to that reference, if anyone is interested. So thank you very much, and I'll take any comments or questions.
Thank you so much, Rowena. Any questions that came across in the chat? Not many at the moment. Maybe more can come through and you can carry that across to Slack. Yes, if people are asking for that reference that to your paper, so if you want to put that into the chat, Rowena, that'd be amazing.
Right. Okay. So I have to exit in order to have access to the chat.
Yeah. Yes. If you just stop sharing your screen and then down on the bottom panel, you should see a little chat bubble, and if you click on that, you can just share the link.
Yeah.
Amazing. Thank you so much.
Thanks.
That was really exciting talk. So now we're all on to the rapid fire stage of our presentations. So we were discussing previously how we're going to handle it if people went over time, because we want to stick to that strict 3-minute time frame. So Albert was talking about getting a air horn happening. I thought it might be a bit abrasive. It kind of, at the end of 3 minutes, it may shock a few people. Oh, sorry.
It's 5 minutes.
Sorry about that. 5 minutes. I've been obsessed with the 3-minute thesis competition. That's all I can think about at the moment when it comes to talking. But sorry, it is 5 minutes. Sorry about that. So at the end of 5 minutes, if you do go over time, you'll get a nice gentle yeehaw like this that will come through and remind you to wrap up your talk if you can the next 10 or 12 seconds. So just to get started, we'll jump on with Siddhant Sharma, and they're going to be telling us about a computational simulation of a reaction network involved in alkaline degradation of glucose. So Siddhant, if you are online, could you please unmute yourself and share your screen?
Yes. Just a minute, I'm sharing my screen. Can you hear me well?
Yeah, we can hear you perfectly. That's amazing. Thank you.
Just a minute. So since I'm doing it for the first time, I wanted to ask, am I allowed to start my video?
Yeah, so you can start your video. If you got some slides to share as well. At the moment, it's just your profile picture that's up.
Okay, that's perfect for me. I don't need fragments, so I'm sharing my screen.
Perfect.
Just keep giving me pointers if you find some disturbances or a Zoom box lying around.
Yep, yeah, no, you're doing well. So you've opened up your a Google presentation, and
Yeah.
It's still not on full screen yet. So if you want to just go to the presentation.
Sorry for taking the time, and I hope I can start by now.
Yeah,
okay. Perfect. So just a little bit introduction about myself. I'm Siddhant Sharma, a biological sciences undergraduate from India. And I did this part of the work as a part of my summer research program with Blue Marble Institute of Space Science as a young scientist program research associate. And my mentor for this program was Dr. Henderson Jim Cleves. He's at ALS at Japan. And so as it's pretty much explanatory, the title of my talk is a computational simulation of reaction network involved in alkaline degradation of glucose. So the main focus of our work was in prebiotic chemistry and origins of life. I was really excited about origins of life since the beginning, as I wanted to find out how the life originated on Earth, how the simple molecules began compiling into complex ones. So moving forward, without wasting any time, I would like to introduce to the workflow of my summer project. And here it is. So we use a reaction network generator known as MOD that was developed by our collaborator, Dr. Jacob Anderson at Amada in Denmark. So I would like to thank him. So what does mod does? Mod is a cheminformatics software that uses graph grammars to generate your rules. So we applied the rules-based reaction network, that is mod, to our already applied compounds. And then we use a bunch of rules. What are rules? So we took tautomers, we applied a bunch of simple organic high school chemistry rules to it to get the outputs. So then we applied various constraints to the output. So as you can see, we got 3,000, 5,000 outputs, and that was not really computationally well. So to reduce down the outputs and to make it workable, we applied constraints such as if the structure is unstable, if the structure has 2 carbons, if the structure has 2 or 3 carbons rings, so it won't be included in the final run. And then we added isomorphism test to match the mass spectra data to the paper we referred to, Yang and Millis, and we studied the alkaline degradation of glucose and formose. That are the main reactions that are involved in my lab. So we studied both of them. So using isomorphism test, we were able to find if our outputs are really valid or not, or if we are going into the great details. So the final part of the topic was checking for autocatalytic loops. I'll come over to what are autocatalytic loops in the further part of my talk, and we used all of this to check for autocatalytic cycles. That was the main part of the stuff that I did for the last previous 4 months. And as you can see on my screen, this diagram, it took me 1 month to figure out how to read that. So that's a +1 for me. And yeah, writing rules. How did we write rules? We took a bunch of molecules. We used tautomers as they are interconvertible, and we use glucose. And we applied a bunch of rules such as shift transformation and 40 different others. And we then feed it up into mod to generate our networks. And then we used RDKit and Ambit to just reduce, figure out, and feed all those tautomers to get the final 30, 40 usable ones. So as you can see on my screen, there is a nice GIF of the procedure. It's had a lot of computational work to do with it. And here's my talk, fantastic autocatalytic loops and where to find them. So how did we model autocatalytic loops and feedback loops? Autocatalysis is really a great topic to study about to study about origins of life, to study about various biochemical pathways and all. So I wanted to say that we aimed for this project, we aimed to build a complete in silico model of the possible chemical reaction networks that could have using automatic computational means. And we hope that this will enable the detection of autocatalysis for understanding origins of life, as I mentioned, for understanding drug pathways, for understanding computational geochemistry and all. And this in silico analysis is very well. So you will see what's the main purpose of our thing. So when my mentor started thinking about it, my mentor went on the board. So what if we had software where it could be pushed a list of input molecules and reactions, and then it could generate all the entire chemical network filled with the chain reaction. It would give us the output. That would be so great. And then the software would analyze and accept the network to give a user with high level, like really a good quality data about the network. So that we did. So we use Neo4j, we use RDK, we use a lot of stuff. We feeded the stuff that was generated by mod into Neo4j, did the graph grammars, did the graph queries. It's a graph-based databases, and then generated a lot of feedback loops. And Neo4j, you can cycle on in a better way. You can generate queries. You can do pattern matching that my other acquaintance did. So that's how we found autocatalytic loops. Moving on. I believe that I completed most of the talk by speaking really fast, but again, sorry for that. So what's the scope of our work in the future next? So as it was a virtual collaboration program with multiple time zones, I was not able to do it that efficiently as I would do in a physical lab space. So how do different reaction networks converge and collide, and how do different autocatalytic loops work together? We need more computational power, we need more manpower for do that. How do thermodynamic parameters like SNA or flux balances and different kinds of stuff to determine the feasibility of our reactions, and how do kinetic parameters determine how our reaction is going to. So thanking all of the people that were in my program, and I'm giving my acknowledgements. And if you have any questions, do feel free to chat me up. I would like to thank my mentor, Dr. Jim Cleves from ELSA Japan, and I would like to thank my collaborator, Dr. Jacob from University of Southern Denmark. And I would really love to talk about my wonderful co-interns, as you can see on my screen. And that's it for me. Thank you, AAM 2020. Thank you all the organization for giving an undergraduate, a really young undergraduate, a chance to speak his heart out on the topic he's passionate about. Thanks.
Amazing. Thank you so much, Siddhant. And you can tell you're very passionate. It looks like really amazing work.
Thank you.
Next up, we have Rajlaxmi talking to us, and they're talking about, do phase-separated lipid membranes enhance the clustering of nucleotides? So as Rajlaxmi, if you could share your screen now, if you wouldn't mind.
Hi. I hope you can hear me.
Yeah, we can hear you fine. It's cutting out a tiny bit, but I think you should be fine. So if you want to take your screen up, perfect. And yeah, whenever you're...
Yeah, okay. So I'll start. I'm Rajlaxmi and I'm from the Srivastava lab in ISC, India. And today I'm going to talk about my work that I did with him, which is titled as the Molecular Organization, Dynamics and the Polymerization of Nucleotides in the Lipid Environment. So a theory that exists for understanding how the nucleotides form prepolymers or polymers is the non-enzymatic polymerization. And various studies previously have shown that non-enzymatic polymerization can take place in various organizing matrix or environments such as clay, mineral surface, or salts. But for our study, we thought we would study the membranes, and especially this unique characteristic that the membrane has, that it can exist in many phases. As in a small range of temperature or hydration, the membrane can change its phase. And we wanted to see if this property of membrane could have any influence on the nucleotide prepolymerization formation. And for this study, we used molecular dynamic simulation and mathematical modeling. Mathematical modeling to understand the problem. So we considered a LO phase, a LD phase, and a phase separated membrane. And for the molecular dynamic simulation, what we did is we took nucleotides and arranged them in ordered arrays. Yeah, if you can follow my mouse here. And then after 200 nanoseconds, we saw, okay, the nucleotides indeed are forming clusters, which I've actually circled them in black circles. And we found these clusters are anisotropic in nature, but we wanted to understand if the nucleotides have any preference for this phase. And due to limitations in timescales of molecular dynamic simulations and evolutionary timescales, we decided we would take a thermodynamic approach. We used advanced sampling methods to do the thermodynamics calculation. Namely, this method is called the umbrella sampling, where we, for the LO and the LD phase, we try to see when the membrane moves, when the nucleotide moves from the solution to the membrane, if there is any kind of preference for the membrane. And what we find is that the nucleotide indeed has a preference for the LD phase if you follow the blue line, which has around 20 to 25 kilojoule per mole energy that it needs to insert itself into the membrane, versus the LD, which is the dark blue line here, which needs almost thrice the delta G to insert through the membrane. So there is a clear preference for the LD phase here. And for the phase separated membrane, we actually tried to study the nucleotides preference across the membrane bilayer plane. And we found that in this second figure here, if the domain interface is somewhere around 1.5 plus minus 0.2 nanometers. So right here, and to the left here is the LO phase, and to the right here is the LD phase. And we see there is a clear minima for the domain interference, so interface. So in a phase separated membrane, the nucleotide would preferably go to the domain interface followed by the LD. And we verified this by a Z distance calculation and by energy calculations. Then in the next step, we developed a mathematical and a theoretical model to understand this. So we took a lattice model, and each point in the lattice is nothing but the lipid in the membrane. And we modeled the nucleotide-nucleotide interaction and the membrane-nucleotide interaction. And what we found is, so this is the starting state here, and the black phase is the LO phase, and the white one is the LD phase, and the blue dots here are the nucleotides. because of the higher diffusion constant of the LD phase, the nucleotides move around a lot, and then they get stuck at the domain interface, and eventually once they move on to the LO phase, they get stuck there. If you can see here in the LO phase, all of the nucleotides are clustered there. So essentially the LO phase becomes a reservoir for the nucleotide polymers or prepolymers. So in conclusion from this work, what we understood is no matter what the phase of the membrane is, the nucleotides are always forming clusters, and they are usually anisotropic in nature. And in terms of phase preference, there is a very clear preference for the domain interface in the phase separated membrane, and there is a preference for the LD after that. And also the LO part of the membrane is somehow being the reservoir of the prepolymers. So somehow we can think the domain interface to be this suitable place which can have the best of both worlds. So they can easily go to this LD phase where it has high diffusion constant of the LD lipids, and whereas if it moves to the LO, the nucleotides are stuck there. So yeah, thank you for your attention. That's all from my side.
Thank you so much, Rajlaxmi. That was great. Thank you.
Oh.
It was really clear, and yeah, it was amazing. Thank you so much.
No problem.
And I hope the cowboy yeehaw isn't too abrasive for everyone presenting. It's the most subtle sound I can find on a soundboard. Next up, we have Lauren Lowe, and she's going to be presenting on regulating the nutrient flow of model primitive cells. So, Lauren, yep, you're set. And could you just say something, Lauren, to make sure your speaker's working?
Can you hear me?
Yes, we can. Yep.
Great.
Amazing.
Awesome. So my name's Lauren Lowe, and I am an honours student working with Anna on regulating the nutrient flow of model primitive cells. So bilayer membranes are vital to controlling what substances move into and out of cells. In existing life forms, cell membranes are composed of phospholipids, and the control comes from membrane proteins, which transport substances into and out of the cells. However, it is not clear how this was achieved in primitive life, prior to the evolution of these complex molecules. I hope to determine how membrane permeability was governed in model primitive cells, providing us with an increased understanding of how primitive life forms regulated their nutrient flow. So bilayer membranes consist of two leaflets of lipids, with a hydrophilic region and a hydrophobic region. Fatty acids such as oleic acid are molecules that can also form bilayer membranes. They are chemically simpler than phospholipids, can self-assemble into giant unilamellar vesicles, and have been found on meteorites, making them ideal candidates for modeling primitive cell membranes. Cells must have mechanisms in place to enable nutrients to pass through their membranes. However, it's not well understood how this was achieved in fatty acid bilayer membranes. One potential mechanism is through passive diffusion, which involves the movement of small neutral molecules across the membrane in accordance with the concentration gradient. Another mechanism is through transient pores, which are holes that quickly appear and disappear in the membrane, allowing ions to pass through. Flip-flop-mediated diffusion is another potential permeability mechanism. This involves fatty acids in bilayer membranes are quite dynamic, so they can flip from one side of the membrane to the other in what is known as flip-flopping. This can transport protons and other positively charged species across the membrane. Previous research has found that compared to phospholipids, fatty acid packing is significantly affected by changes in environmental conditions, such as changes in pH or influxes of salt. So to assess the permeability of fatty acid bilayers, traditional permeability assays which rely on adding additional ingredients to the membranes wouldn't be appropriate. Therefore, I'm using a technique called electrical impedance spectroscopy to directly measure the properties of the membrane itself. It involves tethering a lipid membrane to gold electrodes and applying alternating current across the membrane. This enables the conductance of the membrane to be measured, providing an indication of the ion flux through the membrane. High conductance readings indicate that the membrane is quite leaky. So this plot shows the conductance of oleic acid membranes at slightly different pHs over time, and highlights the sensitivity of oleic acid membranes to changes in pH. The conductance of the membranes was greater at a pH of 8.45 compared to a pH of 8.29. This suggests that oleic acid membranes are leakier at higher pHs. This is possibly due to the increased pH causing there to be more negative charges around the lipid head groups, meaning they repel each other more, take up more space, and favour the formation of high curvature structures, which in turn favours increased pore formation. I've also looked at the conductance of oleic acid membranes that had been cooled in the fridge at 4 degrees Celsius and compared them with oleic acid membranes at room temperature and POPC membranes at room temperature, which are a type of phospholipid membrane. So after first taking the membranes out of the fridge, it can be seen that the conductance was a lot lower compared to the room temperature oleic acid membranes. And as the membranes warmed up over time, the conductance increased. So furthermore, it is likely that when these membranes were cooled in the fridge, they transitioned into a gel phase, meaning that they are more solid-like and meaning that no flip-flopping can occur. Additionally, flip-flopping does not occur in phospholipids over this time frame. This suggests that even at a low temperature, the transient pores are playing a significant role in oleic acid membrane permeability, enough so that the membranes, which already have oleic acid membrane permeability, they're even more conductive than POPC membranes at room temperature, which already have a strong reliance on pore formation as a permeability mechanism. Thus, it can be concluded that even at these low temperatures, transient pores likely play a significant role in permeability of oleic acid membranes. So in the future, I hope to conduct further experiments to confirm the relative roles of the different permeability mechanisms that I've discussed, and continue to explore what other factors govern fatty acid membrane permeability. I'd like to thank Anna for all her support with my project, and thank you everyone for listening.
Amazing. Thank you, Lauren. That was a great talk and perfectly timed. So that was great. Thank you so much. So there are a few questions coming in for this talk and other talks. We may just get to them all at the end. And if we run out of time, we'll move them across to the Slack. So we'll just keep pushing on then to the next person. So now we have, where are we? Sorry, Shota Nishikawa, and they're going to be presenting on screening de novo RNA binding peptides from a sequence space with limited amino acid diversity. So Shota, if you are present.
Okay.
Hey.
Can you hear me?
Yeah, we can hear you fine. And there's your screen.
Okay. Okay, so thank you for giving me this great opportunity. My name is Shota. I'm a master's student at Tokyo Tech, Japan. Today, I'd like to give an overview of my research. Recent study indicated that molecular copulation between RNA and non-coded peptides may have occurred prior to the development of coding system, and it largely contributed to the evolution of the extant ribosome. The close synergy between peptides and nucleic acids in current biology might be remnants of a functional co-evolution at the early stage of life. However, despite the importance of this interaction, systematic exploration of de novo RNA binding peptides has yet been reported. So my research is especially focusing on the early mode of RNA and peptide interaction. Some chemical experiments like Miller and Urey experiments indicated that primitive proteins probably consisted of a smaller subset of amino acids. So in this study, we screened for RNA binding peptides from a sequence space with limited amino acid diversity. And we aimed to target the simplest form of RNA, which is poly A, C, G, and U. Here is the schematics of my research. We used mRNA display to screen for RNA binding peptides. mRNA display is a widely used in vitro selection technique to screen for functional peptides from libraries of more than 10 to the 13th molecules. In this method, you start with a randomized DNA library, and after transcription and translation, we can get the conjugate of peptide and mRNA. The stable covalent link enables selection by specific properties of peptide. And finally, the selected peptide sequence can be obtained by reverse transcription PCR and sequencing. It's a bit more detailed, but we removed complementary base from the codon to avoid mRNA and RNA interaction. For example, poly-G is removed from the peptide coding library sequences, which targets poly-C. And this allowed us to create peptide library that limit amino acid diversity to only 9 types. So far, we have performed 7 rounds of peptide selection against poly-C RNA. And round by round deep sequencing showed remarkable stepwise enrichment patterns for multiple peptides. And surprisingly, the most enriched peptide sequence dominated over 80% of all sequenced reads. And what is interesting here is that according to the rank 1 and rank 2 sequence, asparagine and histidine may contribute to the binding through hydrogen bond or charge interaction. Actually, such a kind of interaction has been appeared in some RNA protein complexes in nature. And so this result is really consistent with the way nature uses amino acids. And this is the first example of successful de novo RNA screening, RNA binding peptide screening. Thank you. And if you have any questions, feel free to ask me.
Thank you so much, Shota. That was a great talk. So we have a few time for questions if there's anything coming up in the chat. If not, we just might push into the next one. And I think we'll have maybe 10, 15 minutes at the end of this entire session to go over some questions again. So definitely keep putting your questions in the chat and we'll have time to address them. So the next talk that we have now is from Daniel Lowe, and it's all around the potential role of asymmetry in the origins of primitive cell motility. So Daniel, if you are with us, could you please turn your screen on and share your slides, please?
Oh, yeah, sure.
Hey. Thanks so much, Daniel. Take it away.
So is it showing?
Yeah, so we can see your screen and you're coming through loud and clear, so that's perfect.
Great. Hi, everyone. My name is Daniel. I'm a master's student from the University of New South Wales in Australia. And today I'd like to share with you my project in which we investigate the role of asymmetry in the origins of primitive cell motility. Cells benefit from the ability to move, which helps them reach nutrients or move away from harmful environments. The motility of individual cells are enabled by specialized organelles, some of which are shown here. However, these are relatively well developed and didn't exist in the prebiotic era. I want to look at how protocells or early cells could have moved without the help of such organelles. In my project, I used vesicles from self-assembled oleic acid molecules to serve as our model for primitive cells. Fatty acid molecules, such as oleic acid, are simpler than phospholipids and may have formed the membranes of protocells on early Earth. Our idea is to encapsulate a catalyst within these vesicles, which catalyzes a reaction, forming a product which will escape from the vesicle's semi-permeable membrane. One such reaction is to use a catalyst to break down hydrogen peroxide to produce oxygen. In order to achieve biased movement, we need to induce asymmetry of some sort to the structure of our vesicle. We have found a way to turn a portion of the membrane more impermeable. This will allow the outflow of oxygen to be directionally biased, which we believe will propel the vesicle. By adding 2+ cations to our oleic acid vesicles, we noticed a portion of the vesicle membrane, which are comprised of fatty acids, may precipitate. Magnesium also happens to be especially important in RNA and DNA chemistry, so it's quite fitting that we use magnesium chloride. However, adding too much magnesium caused our vesicles to collapse, so adding the right concentration is very important. Under fluorescent microscopy, we can observe scarring of the vesicle membrane, as shown here as an especially bright region of the membrane. Under 2-color fluorescent microscopy, we observed a nucleotide. Again, a significantly brighter portion of the membrane indicates where the scarring took place. As a proof of concept, we did try to catalyze the breakdown of hydrogen peroxide to produce oxygen using a protein enzyme called catalase. Unfortunately, this resulted in our vesicles collapsing. And it turns out that oleic acid does interact with catalase. So for us to move on from catalase, we are currently in the process of synthesizing and using platinum nanoparticles as catalysts to see if they would yield better results. In the near future, I'll prepare and encapsulate platinum nanoparticles, which hopefully will propel our vesicles and maintain its structure. We also plan to track vesicle movement and distinguish between the different types of movement using optical microscopy. Thank you for listening to my talk.
That was great. Thank you, Daniel. I've got a lot of very exciting questions for you, so looking forward to chatting about that more later. Next up, we have a talk from Bryan Drake, and it's all around the future evolution of humankind. So Bryan, when you're ready, if you could turn on your microphone and share your screen.
Yes, microphone and the screen should be sharing. How's that?
Yep, so the screen's great. We can hear you perfectly. So whenever you're ready, you're good to go.
Okay, so I'm Bryan Drake. I'm a research associate of Kathy Campbell's at the Auckland University. I'm European and Maori by descent, and I whakapapa to Ngāi Tahu and Ngāti Kuri. Now, we recently had a film showing in Auckland called The Planet of the Apes, and for some reason I'm not wanting to change pictures here. What's going wrong with that? Oh, there we are. Yeah, so humankind has evolved to be a global apex predator. Over the last 300,000 years, we actually became the apes that took over the planet. We did, well, first of all, we stood upright. Yeah, there we are. We got upright nature. It resulted in backache, heart disease, and dangerous childbirth, but it freed up the hands. Our manual dexterity allows us to wield weapons, to thread needles, to make music, and to fly jet aircraft. Secondly, we've got the size right. Small animals, mice, have large body area in proportion to their mass. They lose heat quickly, so they have to eat fast, move fast, breed quickly, and they die young. We are large enough to be able to live long enough to gain skills, to pass them on to our children, our grandchildren, and we're small enough to be agile. Early anthropoids had large jaws. They were good for eating, good for attack. We compensated. Cooking food means we don't get massive jaws, and a small jaw allows a skull with an enlarged brain. So when you're watching Star Wars, forget those weird characters. If somewhere out there there are technological aliens, physics and thermodynamics say they will look very, very much like us. But the brain size is not the only answer. There's a Neanderthal head, skull on the left, and a Homo sapiens on the right. And Neanderthals had larger brains. We humans learned to network our brains by living in society and sharing our thoughts and working together. Now that requires a sense of identity, of common interests, and from early tribal communities, people were cemented by custom, tradition, and myth and religion. Over the last 14,000 years, mankind has moved progressively from tribes to villages to towns to cities and ever bigger cities. And now in the last 200 years, the wealth of a nation has been directly proportional to the percentage of its population living in cities. But that's not enough. Beginning about 1825, the world was transformed into the modern technological society. And Bernstein identifies 4 things that were responsible for that. First, law that protected our property and law that protected our ideas. Secondly, capital markets, money for big projects. Then thirdly, communication, the ability to move people, products, and information. But fourthly, scientific rationalism, the development of evidence-based knowledge. Now, there's a problem. Because the power of science also comes from its reductionism, reducing things to finer and finer detail. So even biology gets reduced to biochemistry, and then to molecular genetics, and then just down to plain atomic physics. And in the process, the holistic picture of the universe is lost. And this is what alienates many. Now, it was wonderful yesterday to have Sarah Jane bringing a First Nations perspective to our studies. In New Zealand, we have something similar with Mataranga Māori. That's where the experience and wisdom of our ancestors has been distilled, codified, and ossified so that it's rather fixed and resistant to change. Even with our Western European traditions, the same thing will happen. We have traditions which have become acceptable and believed and beyond question. It's necessary that we broaden our scientific view to take in arts and culture and traditional indigenous religions and viewpoints, but a problem crops up. This was from Auckland's newspaper, the headline this morning. We have a little outbreak of COVID-19 in Auckland that centers around a group of Christians who refuse to believe that the COVID virus really exists, and they won't social distance, and they won't get tested, and the virus is spreading amongst them. Now, there's a minority of all those who go to church, but the same sort of problem crops up with Mataranga Māori and with some of our traditional viewpoints, because all too often we have people who will not adapt and adjust their traditional beliefs and alter them to take account of what is evidence-based knowledge. In Maori tradition, there were 3 kete, 3 baskets that Tane brought down from heavens to earth, baskets of the world, of good and of evil. Our ability to survive depends on us taking our own kete, our own basket, and putting into that our traditions, whether they're Western or whether they're from original native traditions, or wherever they come from, and putting that in along with our science and our insistence on evidence-based knowledge. Daniela this morning had a talk about her Navajo and the way they had a foot in 2 worlds. We're aiming for the same with our Maori people in New Zealand. But all of us have this need to merge our science with the wider traditions around us, but we still have to make sure that at the basis of that, we're depending on evidence-based knowledge and not allowing ourselves to be crippled by people insist on limiting and that by obedience to what they have accepted by tradition.
Sorry, Brian, I just accidentally muted you. Can you please just keep going for the last 20 seconds of your talk?
I had finished.
Oh, wow. Amazing. Thank you so much. That was great. So I'm sure there'll be a lot of talk, a lot of discussion coming out from that, Brian. It was really insightful, and really, ties into the holistic approach that we've tried to embody in this presentation. So we've got time for a few more questions at the moment, and we'll just jump to the first one that I could see, which is coming to Daniel on his motility in the lipids. And that's all around, how do you minimize the lateral diffusion of the lipids in the scarred region of the bilayer? So Daniel, if you want to comment on that, that'll be amazing. if you
Oh yeah, hi. I think I typed the response, so it's
Okay,
sitting there.
Yeah, no, thank you so much. Amazing. So we have a lot of interest in all the talks coming through. So it looks like most of the questions have been answered in the chat. Does anyone have any questions they want to ask, either in the chat or live? They want to turn the microphones on and have any chats or comments. I know Brian's talk was especially thought-provoking, so if anyone has any reflections on that, we'd all love to hear it.
Hi. Yes, I've got a question for Brian.
Yes.
I mean, it's more like a reflection. Like, I find that here in Australia, a lot of people are really not receptive to acknowledging and respecting native Australian culture, and like they struggle to like take it into account in policies or things like that. And I wonder, how is it happening in New Zealand? Is there a better acceptation and integration of native communities into policies?
Well, I think in New Zealand we have had the advantage because we had a treaty, the Treaty of Waitangi, and although that has been a contentious development over the years, I think we've made much more progress than in Australia. This week is actually Maori Language Week, where it's being emphasized. There's been a lot of use of Maori language in normal conversation. There still is quite a bit of reaction. You will always get people who react to anything that's strange coming in, and it's a work in progress, but I think we're getting on better than you have in Australia.
Thank you. And I might just add as well, for those of us in Australia who are interested, there was a meeting a few years ago at Uluru, which is the big geological rock in the middle of Australia. For those who aren't aware of it, it's a very sacred site. And they had a meeting of Indigenous people all around Australia getting together, and it was called the Uluru Statement from the Heart. And they really unified and came together and said, we want a treaty in Australia as well, and we want to be recognised as Indigenous people in the Australian Constitution. So I think there is a movement for a treaty in Australia as well, but it's definitely slow going compared to the amazing work that so far has happened in New Zealand, as well as all its difficulties I can imagine. But there's definitely a lot more progress from what I can understand. Thank you for sharing, both of you.
Yeah, so it's a work in progress, but it is progressing. That's quite certain.
You know, thank you, Brian. Is there anyone else that has any questions or comments I would like to share with the group?
Just perhaps another comment on the New Zealand situation. We're heading into an election. In New Zealand, we do have Maori MPs. So well, if you're Maori, you have the right to be on the European roll or on the Maori roll. And so we are guaranteed 4 Maori MPs in Parliament elected from those who are on the Maori roll, as well, of course, of having Maori who stand in the ordinary electorates.
Yeah, no, that's amazing, and definitely something that we don't have here in Australia. So hopefully we can take a leaf out of your book and try and sort of, instill a bit more, I guess, autonomy and direction in our own Indigenous people in Parliament. So yeah, someone's made a comment that there's a journal called People and Nature that particularly focuses on Indigenous and traditional knowledge and their connections with nature. So that could be a really great journal for people to check out if they're interested. If we have no more comments or questions coming through, or any more people who want to make a bit of a reflection on what's happened, we may wrap up this evening session, this afternoon session, and we'll see everyone tomorrow if that's it. So thank you all so much.
Fine, thanks, Luke.